Now we're going to look at protein synthesis. We've got our model of DNA, and we'll show the two steps involved in making a protein. The first step is called transcription. And what happens during transcription is that the DNA molecule opens up along a particular gene. So the molecule opens, and messenger RNA, which is colored red or pink on my models that you'll see in a minute, messenger RNA will come in and bond along one side of the molecule. So I'm showing the messenger RNA coming in right now, and it is basically copying the code as it bonds up to one side of the DNA ladder. So this strip that I just made is called messenger RNA, and I don't know if you can tell, but it is a pink color. It may not be showing up. Messenger RNA is similar to DNA in that it's made up of a sugar, an acid, and a base. However, the sugar is ribose, and there's one base, uracil, that is not found in DNA, replaces thymine. So back to our story here. Messenger RNA has just copied the DNA code. Copied the DNA code by bonding up to it. That is transcription. Next, that messenger RNA will detach and it will go to a ribosome. So we're going to try to show that. I'll move it away. It's going to detach from our DNA, and it's got to stay intact. So it's going to chug its way out of the nucleus. We're going to pretend that it's now out of the nucleus, and we will suppose that it has just arrived at a ribosome. And our DNA, in the meantime, is staying in the nucleus, of course, and it's going to close back up. So let's move this out of the picture. And my messenger RNA supposedly has arrived at a ribosome. All of the enzymes are present at the ribosome, so here we are at the ribosome. Now starts translation. For translation, we've got transfer RNA that's going to be involved, and each transfer RNA is made up of 3 bases in a row that we call anticodons, and each tRNA is carrying a particular amino acid. Here's another transfer RNA, and it's carrying a different amino acid. So this molecule will bond wherever it is able, and this one happens to bond right here. And the next transfer RNA will transfer the next amino acid. Now imagine that this chain is actually hundreds of bases long, and many different transfer RNAs will be coming in and dropping off a different amino acid. And as each transfer RNA drops off an amino acid, the lower part of our tRNA, I call them Tonka trucks. The Tonka trucks go out into the cytoplasm, and they will go and search for another amino acid to bring. In the meantime, these two amino acids are bonding together. This transfer RNA goes out to the cytoplasm to go fetch another amino acid, and you can see that you are beginning to get a chain. We've got 2 amino acids bonded together. We'll get more and more and more, and that will create our protein. This will become the protein once we have more and more amino acids bonded. Once the protein is finished, the messenger RNA will simply break apart, and the nucleotides will flow back into the cell. And we'll be left with our particular protein, which will be folded and exported to wherever it needs to be.